Hello, I'm Shreyas. I'm going to be teaching you DNA replication and protein synthesis. The first thing that we're going to be talking about is DNA. DNA stands for deoxyribonucleic acid. DNA carries genetic information, and DNA is found in almost all living things. DNA has a double helix structure, meaning that there are two strands running in opposite directions that coil up. The two strands are made up of nucleotides, which is a molecule, and nucleotides are put together to make two strands. The outside strands are known as sugar phosphate backbones, and in the inside, the ladder-like structure inside, are the bases. There are 4 different bases inside a DNA structure. The 4 different bases are cytosine, thymine, guanine, and adenine. Now we're going to break down the DNA structure part by part. First, we're going to talk about nucleotides. Nucleotides are the two strands that make up nucleotides are the molecules that make up the two strands in the double helix structure, like I said before. But there are 3 main parts of nucleotides: a phosphate group, 5-carbon sugar, and nitrogenous base. Nitrogenous bases. The 4 nitrogenous bases in DNA are the building blocks of DNA. The four types of nitrogenous bases are cytosine, guanine, thymine, and adenine. The 4 bases are paired up, meaning that cytosine goes together with guanine, and thymine goes together with adenine. The two bases are linked up by carbon bonds. Adenine always pairs up with thymine. Adenine always pairs up with thymine, and cytosine always pairs up with guanine. So in a DNA structure, the strands would look something like this, with adenine and thymine pairing up with hydrogen bonds running through it, and cytosine and guanine paired up. Hydrogen bonds are weak bonds in between 2 molecules that creates an attraction between the two molecules. The reason why hydrogen bonds are weak is so DNA replication can happen, meaning that the DNA structure can unzip easily and make two new strands. Hydrogen bonds are also present in nucleotides. Replication is the process of making identical copies of the original. This occurs in the nucleus. DNA replication is making 2 identical strands from the first strand. The 2 strands split up at a certain location known as the origin. And this location makes a fork in the original DNA, splitting up in 2 different parts. First, the helix uncoils in a specific location, like I said, which is the origin, and then proteins and enzymes work together to make the new strands. Lastly, an enzyme called DNA polymerase assembles the new strands, making the 2 identical DNA structures. Now we're going to be talking about transcription. During transcription, DNA is used as a template to produce RNA molecules. There are 3 main stages to transcription. The first stage is initiation. This is when RNA polymers bind to the promoter, which allows DNA to unwind and be read. The second stage is elongation. This is when this builds the mRNA using the coding in the DNA, and complementary bases are still present in this stage, but the adenine is paired with uracil instead of thymine. The third stage is termination. This is the end of transcription. mRNA is done and detaches from the DNA. DNA polymerase knows when to start and stop coding the DNA because of the codes known as codons. The start codons inform the sequence to start, and the stop codons tells the sequence to stop. Transcription happens in the nucleus. Alright, from this picture we can see this is the DNA. This is the DNA strand, and in this stage it's split up into two, meaning there's one coding strand which is red, and there is a template strand which is used to create the RNA using the coding from the coding strand. So after it has been coded and read, and it's made into the RNA. After the mRNA is made, it leaves the nucleus and goes to the ribosomes in the cytoplasm. This is just the templates. Another picture for the templates. All right. After the mRNA, like I said, it goes to the cytoplasm, and that's when translation starts. Translation is a stage that happens after transcription. Translation is when the mRNA from the transcription is decoded to make specific amino acids/polypeptides. In other words, this is where proteins are made. The first step of translation is when ribosomes attach to the mRNA molecule in the cytoplasm, using the start codon AUG. Then the codons are passed through the ribosomes like a conveyor belt. Then the tRNA gets the right amino acids attaches the right amino acids to the right ribosomes. Then the ribosomes attach to the amino acids, making a chain. Every tRNA has a anticodon, which are three unpaired bases. Each anticodon is complementary to a mRNA codon. The two main parts of tRNA are the anticodon and the ancestors. RNA are the protein builders of the cell. rRNA translates mRNA into protein. They make protein by joining amino acids in the right order. It knows where to stop and start translating because of the start and stop codons. The start codon is AUG, and there's only one of those. But the stop codons, there's three stop codons, which are AUG, UAA, UGA. Okay. From this picture, we can see that the tRNA brings the right material needed for the mRNA that is brought from transcription. It's like a conveyor belt, because this is the belt, and the materials that are needed are brought and attached to the bases, and then it leaves and then starts another cycle. But this is where the protein chains are made out of these, and the amino acids are made. This shape right here is the ribosome, and that's where it happens. That's where protein synthesis happens. Polypeptide bonds are covalent chemical bonds that link two amino acids together. They are found in amino chains and polypeptide chains in a protein. Mutations. Mutations are mistakes in copying DNA. This could be inputting the wrong base or skipping a step in a sequence. Mutations are heritable changes in genetic information. There are two types of mutations: chromosomal mutation and gene mutation. Chromosomal mutation is a change in number or structure of chromosomes and can change the number of genes. There are 4 types of chromosomal mutation: deletion, duplication, inversion, and translocation. Gene mutation, on the other hand, is a smaller mutation than chromosomal mutation. Gene mutation is a change in a single gene. These are the types of chromosomal mutation. There's deletion, where a sequence gets deleted. There's translocation, where a sequence gets mislocated. And there's duplication, where a part gets duplicated onto another whole part, leaving this part empty and this part with an extra part. Finally, there's inversion, which is the last chromosomal mutation, which is the change in a part between two sequences. Mutations can cause harmful or helpful effects. For example, mutations can cause genetic disorder and can cause a sort of cancer. On the other hand, mutations can help insects resist chemicals and help humans better adapt to an environment. Humans or insects better adapt to an environment. Scientific and technological innovations in DNA discovery. DNA discovery can benefit us as a whole by allowing scientists to better understand what we are made of and how our body and cells operate to maintain our homeostasis. DNA discovery can lead to cures to more diseases and decrease mutations in the future, maybe. An example of DNA discovery being beneficial is by allowing doctors to understand what is wrong with the patient by understanding their cells and their DNA. That concludes my lesson on DNA replication and protein synthesis. Thank you.